Okay, in today's lesson, we're going to do a quick review about our 3 biomolecules we've covered up to this point. We talked about 2 reactions. One is where you are forming, and one is where you are breaking down. Whenever we are forming something, making things more complex, making bigger chains, making a disaccharide, making a dipeptide, making just a polymer, which is just a very general term, we use the process of dehydration synthesis. And this is going to be consistent with all of them. On the contrary, whenever we are breaking down, we need to add that universal solvent, that good dissolver. And that is going to be hydrolysis. And that is consistent the whole way down. Now, in further breaking down what happens with each macromolecule, carbohydrate simply means carbon and water. So what elements do we contain here? We have carbon, hydrogen, and oxygen. And look, that's how we get that 1:1 ratio, as we see with the C and then H2O. The building block or monomer of a carbohydrate is going to be a monosaccharide. And if you have not figured out yet, we are on page 16 to review within the Unit 2 workbook. Functions and uses of carbohydrates. First and foremost, we have quick energy. You also have structure. For both plant and animal cells. Our examples we'll talk about from the polysaccharide standpoint. You have your starch, glycogen, and cellulose. These are all polysaccharides. And if you want to talk about a monosaccharide, remember our glucose. That's our mono. Lipids, on the other hand, they also are going to contain carbon, hydrogen, and oxygen. However, they do not have that distinctive 1:1 ratio. The monomers are building blocks. They do not have a true monomer. Our 3 types of lipids have 3 completely different shapes. What lipids are used for? Long-term energy. Structure. Remember, cell membrane here. Whereas your cellulose as a carbohydrate is going to be your cell wall of your plant cell. Your lipid bilayer is another name for your cell membrane. So we have those phospholipids. And then we also have chemical messengers. These are going to be like your hormones, your steroids to help you out. Examples, we of course have our triglyceride. You have your phospholipid. And because of the phospho part is why I'm going to put P in parentheses, because that's the only one where we see phosphorus. and then we also have our steroid. Moving along, we also have proteins. Proteins have carbon, hydrogen, oxygen, just as the other two do, but they also have nitrogen and sulfur. The monomer of a protein is going to be an amino acid, in which case we have 20 of them. Functions of your protein. Structure. Once again, think of your muscles. We also have transport across the cell membrane. And then arguably one of the more important ones, your enzymes. And remember, your enzymes speed up. I'm just going to put RXN for reactions. Our examples, obviously, your enzymes again. And you'll also hear the term polypeptides. Some key things to remember: your proteins do have what's called your peptide bond. Your lipids are going to be nonpolar. And we already kind of broke down the carbohydrates name to help you out with that. Before we kind of talk about nucleic acids, let's at least spend a couple minutes kind of going over the specifics with that. So, nucleic acids. Think genetic information. We don't really talk about this with food because the majority of your food has it, so we don't necessarily have to test for it. So we're talking about genetic information, primarily that's going to be our DNA, and carrying instruction for building protein, that is going to be our RNA. Just as with the others, we still have that CH and O, but we once again see phosphorus again, which we haven't seen since phospholipids, and we also have nitrogen. The building block of a nucleic acid is what's called a nucleotide, and there are three parts to it. We have a phosphate, and this is where we see that phosphorus at. We have a sugar, which we do have a carbohydrate that makes up a nucleotide. And then you have a nitrogenous base. And why is it called nitrogenous? Because it has nitrogen. We have a whole bunch of nitrogen in here. Three parts make it up. When we talk about the double helix, the sides of the ladder are going to be your alternating phosphates and sugars, and then the rungs of the ladder, like you would put your feet on, are going to be your paired bases. So whenever you pair up your nucleotides, you're making up the strands, and then once you bond the strands together, that's what makes up your double helix. So we have two major types. You have DNA. The NA stands for nucleic acid. And you have RNA. What they are named after is their sugar. Deoxyribose is the sugar that is in DNA, hence why it's D for DNA. When we look at DNA, we have two strands, and the bases are bonded to put those strands together. Our base pairings that we have with DNA, you have A, which is adenine, bonds with T. So A and T bond together. And then you also have C, cytosine, bonds with G, guanine. They bond together. Base pairing rules. On the other hand, RNA, similar yet different. It has its own sugar called ribose sugar, which is where it gets its name from. It only has one strand. And the reason it only has one strand is because DNA is too big to get out of the pores of the nucleus. So RNA, being one strand, is small enough to get out of the nucleus and get the message out to build proteins. So being single-stranded serves that purpose to get the message out. We have similar base pairing rules in that we do have C bonding with G as before, but we have a new nitrogen base in RNA, and that is called U. We still have A, but it bonds with U because RNA does not have T. So A bonds with U. So the biggest thing when you think of genetic information, think of your nucleic acids, RNA, DNA. We will be spending a lot more time kind of understanding kind of what function does that have in the building of proteins, and how that kind of works. How is a protein made from the instructions of DNA? But for right now, it's just kind of understanding just the basics in regards to what elements are around, what's the building block, and kind of how does it compare to our other three biomolecules. So that kind of makes the end of our information. So let's revert back to our big table and kind of finish it off. So we still have our C, H, and O, but we also have nitrogen and we also have phosphorus. So the only element we do not have with nucleic acids is sulfur. Our monomer is going to be the nucleotide, which it has same basic name, nucleotide, nucleic acid, kind of easy way to remember that. Its function is going to be genetic info or carrier. Of genetic info. And we have just our two big examples, DNA, RNA. And your nucleic acids, just a kind of a big thing, they are named after their sugars. That's where we get DNA for deoxyribose, RNA for ribose. All right, that marks the end of Unit 2 material. Let me know if you have any questions. Thanks for watching.